Institute of Clinical
Recording is on.
Clinical Molecular Biology at Kiel University. And this is situated at the medical faculty. So I have a professorship at the medical faculty. But from my training, I'm a biochemist and an archaeologist. So I always try to combine more or less both fields. And our ancient DNA is as well dedicated to this idea: what can archaeology or the ancient remains contribute as well to modern disease and modern medical questions? Maybe as a kind of background for the ancient DNA research, if somebody didn't heard of it until now. So we can use a lot of different biological material where DNA or RNA is encapsulated and survived over time. So this is the classical material, as you see in the upper left corner, for example, skeletal material. We have new materials like the fossilized biofilm, so the so-called dental calculus in the middle upper picture. We have as well exceptional well-preserved organic material like the frozen ice mummy Ötzi here, but as well other materials became more and more important for different questions which we can apply. This is, for example, parchment, which is made from animal skin, or paleofeces, which we can find as well in archaeological records, or frozen soil material, which is as well an interesting resource if you want to reconstruct, for example, the ancient environments. An ancient DNA sample is always a so-called metagenomic data set, so a mixture of different things which are encapsulated in this biological material. It can harbor, for sure, as well, things which are influencing the human individual during lifetime. So it could be the nutritional basis symbolized by the small mammals, or the pathogens which are infecting the persons, which we can as well see and detect in our data. A big issue is contamination, so especially over the preservation time and as well during exploration and conservation, a lot of things are invading into the bone matrix, for example. That's why we are working in so-called clean room environments to keep additional contaminations really low. And here are some pictures of our clean room in Kiel, where we have installed a special filter system where we have UV radiation overnight to decontaminate every surface, and where we can take the sample in a safe and clean environment. Afterwards, we're converting always these DNA extracts in so-called high-throughput sequencing libraries, which are then sequenced on these platforms. And in the end, we get millions and trillions of sequences from these machines, which we then have bioinformatically sort through the references. As symbolized here, we never can reconstruct full genomes. So the genomes harbor, for example, repetitive areas, which we cannot cover with our short and damaged sequences. How you can think about a metagenomic mixture is a bit like you have a pile of different papers, for example, or newspapers here. In this example, you shred it over time, you're losing half of it or more even, and then try to reconstruct from this pile of really fragmented newspapers to reconstruct one particular edition of this newspaper. And you want even to go into the newspaper itself and read some of these articles. And this is the problem of the bioinformatics, to really sort back all these small fragments of the mixture to one particular issue where we want to do, for example, evolutionary studies, or to see in the function of different genes. While we are doing this, a lot of manual work and really cost-extensive work, it makes us able to do a really kind of time travel. So we can go back to certain moments in history where we can analyze, for example, extinct species, or as depicted here, or disease when they are in an outbreak and evolving quite quickly. And to symbolize as well the difference between modern and ancient DNA, this is depicted here. So up is the modern DNA, which we can extract, for example, 1 microgram DNA from 1 gram of blood in modern DNA. And what we can isolate and analyze are really long fragments of DNA. And they are really suitable for all the modern diagnostic techniques we have in hand. And contamination depicted here in red, they are of a minor importance. So they are there for sure, but they are not playing a big role in modern diagnostics. If we go to ancient DNA, we have roughly 100,000 times less DNA in our sample material. And we are dealing always with these so-called metagenomic data set. So we have as well to the blue ones, we have a lot of green small fragments, we have a lot of red fragments, which are more or less presented as well or invading into the bone matrix, like the plant material or the microbiome of the soil background. We are dealing as well with highly modified DNA. This is symbolized by the colorful small things in these blue fragments. And this is the fact that DNA is degrading over time. So we have a lot of chemical and physical modification, especially in the ends of our DNA fragments. And as you see, the DNA fragments are really, really short. So they are shorter than the contaminations and they are shorter than the environmental DNA, which we normally can extract from our DNA samples. That's why the ancient DNA is always performing less good in later on analytical steps. Our lab is, as I said, mentioned, specialized or on more medical approaches, and there are two big hypotheses out in the modern medical world that the infectious pathogens are really old and most likely are originated always in Africa and spread more or less with these out of Africa movement of the Homo sapiens and spread therefore all over the world. This is one paradigm which we have worked on a bit. And the other paradigm is that we have a quick, or this red queen hypothesis, where we have a constant evolution between pathogens and humans, where pathogens are influencing the human immune system. And as soon as the human adapted more to the pathogen changed again their ability more or less to infect humans and so forth and so on. So we see a kind of up and down between pathogens and the human host evolution ongoing. So these are the two big hypotheses we are working on and where we wanted to get more insights from ancient DNA to see really when pathogens evolved, when they are spread to the human population and became really this devastating disease, which we know, for example, from the medieval time or the early modern days. I want now to introduce you or bring you back into the Middle Ages, where we did a couple of studies and where we see for the first time an increasing population density, especially in Central and Northern Europe, where we have the first urban settlements established, where we know that a long distance trade is well established and that we have a big exchange, for example, via the Silk Route. And we can assume that we don't have the same hygiene measurements like today, but we still have a pure hygiene in this time frame. So a lot of potentials that new disease can develop and circulate in a population. The first example I want to present is leprosy, so-called Hansen's disease. It's a complex chronic infection of especially the nervous system and the mucosa. It's a slowly developing disease, so it has an incubation time from up to 30 years. It presents in macrophages, monocytes, and Schwann cells. That's why it's affecting as well first the nerve systems more or less, and can therefore cause as well long-term damage of these cells. You see as well down there that we have around 200,000 new cases each year, and especially in certain regions of the world, here depicted India, for example, or Brazil. These are hotspots still for leprosy infections where we still have problems to cure and treat this disease. It's interesting that leprosy have various clinical manifestations. It can go from mild skin symptoms or lesions up to really severe deformations of the skeletal material even, which can be of use in archaeological terms. And this is mainly due to the host response. So we know that the disease is really, or the phenotypes are really dependent more or less on the strength of the human immune host system. If we go back in time and have a look on the European history, we know that leprosy was endemic between the 11th and the 16th century. And we know from anthropological records that around 5% of the skeletal material really show specific symptoms for leprosy. Knowing this more or less, we can calculate that most likely everybody in this time frame came into contact with the leprosy bacterium, so that 100% more or less of the persons were infected with leprosy, but didn't show really symptoms of the disease. Another interesting fact is that leprosy disappeared from Europe in the 16th century without knowing any treatment. So we didn't have antibiotics, for example, in hand. We know that there are sporadic smaller outbreaks of leprosy in certain areas of Europe, but the majority of these outbreaks vanished more or less in the 16th century. Another interesting factor is that, different to other disease, the people were really ostracized, so they were not part of the community any longer as soon as they diagnosed with leprosy. And that they built in this time the so-called leprosaria, which are the precursors, more or less, of our modern-day clinics. So we see a hospital system more or less developing based on this epidemic outbreak of leprosy in the medieval time. I brought here some cases and controls which we used. So interestingly, leprosy can really be diagnosed on the bone material, quite robust. So you see on the left-hand side a really severe case of leprosy, in the middle a mild case of leprosy, and on the right side a control case, so who had not been affected by leprosy. And you see in the two cases that leprosy really affects the mucosal area and the nerval system. So the loss of the nasal small bone fractions are going on in the severity, and the loss of the upper maxilla is another good sign and indicator for leprosy. We had the luck to work with Danish colleagues in one of the few leprosariums which are excavated. And you see here on the number of graves, 1,500, that we have had really here the chance to acquire a lot of sample material and to do bigger studies, which are not really common in ancient DNA approaches. There it was always the case that single individuals were known with leprosy infection, but here we had really the chance to go in a broader sense to do really kind of case control studies as well. The Danish colleague, the anthropologist, did already a lot of epidemiological studies and could show that the incidence rate of leprosy is declining really in the 15 and 1600s in Denmark. And this is visible not only in this leprosarium, but as well in normal cemeteries which are surrounding this Odense leprosarium. And therefore, we had a good basis to start with our ancient DNA approach. The first step was for sure to sequence the whole leprosy genome to see if leprosy itself changed over time, or if we see virulence genes which are more present in this medieval time, which could represent, for example, this epidemic outbreak. And what we can show here in an early study is that the leprosy genome in the medieval time was not really different to the modern day variability. So we see every SNP and every variation, which is depicted here on the right side. In modern strains, these are not the highlighted ones, and the red ones are the ancient ones, is as well present more or less in medieval time already. Later on, upcoming study from our group is this one here, where we sequenced many more sites and many more medieval leprosy genomes, which are depicted in the upper figure. And there we see already a really colorful picture, more or less, of leprosy in medieval time. In the lower picture, you see the modern day diversity of leprosy. They are divided in so-called branches from 1 to 5, or from 0 to 5. And you can see that they have a certain origin or geographical distribution in modern days over the world. That's why it was always claimed that leprosy was brought out of Africa, more or less, and distributed therefore over the world in different regions and developed own strains, more or less, in different parts of the world. What we can see now in medieval Europe is that most of these colors are already there, and that we see as well a lot of different strains, more or less, represented in a single position. And this is, for example, the leprosarium in Odense again, where we did this large-scale studies and see a lot of these strains already represented in Central Europe. Therefore, we can hypothesize that Europe played a big role in the diversification of leprosy in the medieval time, and from there on as well, the spread of the different leprosy strains all over the world. So we can bust more or less this hypothesis that leprosy was really old. And coming out of Africa, it's more that Europe played really a dramatic role in the dispersion of leprosy. We can even back calculate the most recent common ancestor, so the grandfather, more or less, of the leprosy bacterium, and it's just dating for 4.5 thousand years ago. So it's just developed more or less in the late Bronze Age, early Iron Age, most likely, and spread from there to Europe. We had a look as well on the host immune system, if this played a role maybe in the 16th century that leprosy declined. As I said, we had the luck to examine a lot of cases from the Odense leprosarium, and these cases are depicted here in red. We examined as well a lot of control individuals which didn't show any symptoms of leprosy from the same time frame. And we compared it to modern day frequency, which we had from databases. And we focused in particular on one HLA variant, the HLA-DRB1501 allele, which is known from modern day genome-wide association studies, which makes people more susceptible for leprosy. And we could really show here for the first time that case control studies worked in ancient DNA, and that we identified the same risk variant, more or less, also in medieval Europe to get leprosy. Interesting is that this variant is still quite frequent in Central Europe, which points to a balancing selection on this HLA allele. And we know that these alleles as well involved into the development of MS, so multiple sclerosis today, which is interesting. So we can thank you, the leprosy patients, more or less, that our risk today is reduced for MS. We did then another big study in the Heilig Geist Hospital in Lübeck. So this is one of the few big mass graves which are uncovered from medieval time. Interestingly, from the archaeology, it's dated into a plague pandemic outbreak phase, so in the time frame of the second plague pandemic. And therefore, it was always claimed that it's really a plague pit. And we tried to follow this up and to identify maybe some individuals which are plague carriers. In total, we screened 103 individuals, and in the end, we identified 10 positive individuals which are carriers of Salmonella enterica paratyphus C, which is causing typhus as a phenotype or a disease. And interestingly, they are clustering really close to other medieval known Salmonella enterica strains from Norway and Mexico as well. So again, here, it looks like we can contribute with our ancient DNA technique to the diagnostics of these mass graves. And we can claim that this mass grave is most likely due to a typhus epidemic outbreak and not to plague. And that Rio played a major role in the diversification of these Salmonella enterica strains, which are not any longer a big issue in modern day Europe. Coming actually to the plague, it's important to remember that the plague is a typical zoonotic disease, so it's not a primary human disease. It can be effectively spread to the human, but you always need the primary host, so the rodent populations, and you need the flea as a kind of vector to be transmitted in the human population. We know that or Yersinia pestis caused 3 big outbreaks and pandemics, and these are the biggest pandemics we know, or these are one of the biggest pandemics we know from history times, at least. And I will go more into the detail of the second plague pandemic, which started more or less with the Black Death, so first phase, where a lot of people died in Europe, up to 50% are estimated. And then are reoccurrent until the 16th, 17th century, where we see smaller outbreaks in the second plague pandemic. As I said, there was the first outbreak, the so-called Black Death waves, and you see on the right side the distribution or the time where Black Death or the Yersinia pestis is circulated in Europe, and you see a succession more or less from the Mediterranean area to a more fringent area in in Europe. Interesting is as well that Yersinia pestis caused these sporadic outbreaks until the 17th century, and it could not be explained up to now why it's getting more sporadic and not to these drastic pandemic events like in the Black Death waves, so in the first couple of years. We had the luck to investigate a multiple burial from Riga, Latvia, from the 17th century, so in these later outbreaks, in the sporadic outbreak phases. And they are falling into the family tree of Yersinia pestis, quite developed in this branch, where we have a lot of second plague pandemic strains already visible. So for this, it was quite clear that we see a further developed Yersinia pestis strain, more or less, branching off of the initial death strains, which are originated more on the basal lineage, like these London strains here, which are resembling the Black Death strains, and they are a bit further down, more or less, in the development. We then came across a small region in the Yersinia pestis genome, and in this genome, there was the PLA region located, which is quite important for the effectiveness of infection of Yersinia pestis. And we detected this strange pattern here in the genome. So all these small grayish lines are representing true sequences which we acquired from these individuals. And you clearly see that some regions are really well covered on both sides, and that is exactly this region around the PLA gene is somehow depleted. So we see only few reads which are covering. We reconstructed then an artificial sequence or reference sequence where we try to see if there are reads which are starting in the really well-covered sites on the left side and ending more or less in the well-covered side on the right sides, which could be an indicator that this region is completely missing or deleted in this genomes. And you can see it here simplistically that we have a lot of reads which are somehow which are really bridging this gap. So we have a lot of reads which are starting with A and ending with B, and lacking really the C region, which are represented more or less in this PLA. Good to know is that PLA is allowing the fast spread over the lymphatic systems and the growth of Yersinia pestis in the lower respiratory tract. So it's really one of the major factors which makes Yersinia pestis really deadly and virulent, more or less, to human population, and causes as well the fast outbreak and develop of the disease itself. So we can really show that our strains in Latvia had a depleted region or PLA region, and therefore most likely were not that virulent like the before established genomes from the Black Death strains. So we then tested as well other post-Black Death strains, which you can see here on the left side, which were published. And all of these strains had the same pattern. They had a depleted region in this PLA area. So we can see here really ongoing development of Yersinia pestis, or an evolution to become more avirulent. So they are less virulent in total, and could maybe as well explain why we see only sporadic outbreaks in until the 17th century. Interestingly, there are for sure other measurements why Yersinia pestis slowly vanished from Central Europe. The pest bonnets are a kind of fake from some of the people which are now known from historian, but we had other effective measures like, for example, isolation and quarantine, which are developed as well during the second plague pandemic. Jumping a bit further back in time to the Neolithic period, you may know that this is defined more or less as the period where we have the first appearance of domestic animal and plants, and where we switched from hunting and gathering to a sedentary lifestyle. And therefore, the ability or the events are becoming more often that we can deal with zoonotic disease. So we're living close together with domestic animals and more animals in a more crowded space. We have as well most likely to do with environmental pollution, as depicted here down in the picture. So we have a lot of circumstances which are comparable to the modern day lifestyle, and where we see as well the origins of some of our traits which we are still dealing with today. We had the luck then really to find up to now the oldest published Yersinia pestis genome, which is dating roughly around 3,300, 3,500 BC. It was detected in an individual from a Latvian site called Rhinocans, shown on the right side, where we have examined mandible, where we extracted Yersinia pestis. Up to now, there is a couple of more genomes accomplishing this picture. Two from the site from Warburg, a couple of more from a site in Göckem in Sweden, and we found or detected as well in published data an infected dog from the island of Gotland, where we can as well show that the dog was carrier of this early Yersinia pestis strain. If we have a closer look in the phylogenetic positioning of these early strains, we can see that they are broadly divided into two classes. So in the so-called LN, so in the late Neolithic strains, where as well our strain from Latvia and the Swedish and the German strains are falling on the dog, and a lot of strains which are called LNBA, so late Neolithic Bronze Age strains, which are existing over 2,000 years and spanning more or less over complete Eurasia. The interesting fact is that the LN strains, so the late Neolithic strains, the early ones, are quite diverse. They have long branches, and they're situated somehow at the bottom of the development of Yersinia pestis. All the later on big outbreaks, so the first, second, and third plague pandemic, are situated here in the upper part of the branches. And they are developed, they are in their own development, which are branching off before these LNBAs. In the LNBA clade, you see as well a development more or less, or an adaptation to something which we don't know up to now. And interestingly is as well that all these strains from LNBA and LN are lacking a gene which is important to be transmitted, or that makes Yersinia pestis be able to be transmitted by the flea vector. This is so-called YMP gene, which is still lacking and be acquired as soon as you see the branches appear for the later big outbreaks. They can always be spread by the flea. As I said, most of these cases are really spread all over Eurasia, so we don't know the origin actually of this disease. And from the number of cases we know up to now, so there's a couple of more since 22, but still we have maybe 40 cases over 2,000 years. So this means roughly calculated every 50 years, one case in Eurasia of Yersinia pestis in the moment as infection rate. As I said, they're still lacking a couple of genes, which makes these early strains really virulent. This is, for example, the YMT gene, and later on some modification in the PLA genes, which leads to the modern day strains. We don't know which origin or which rodent host really was able to spread more or less these early strains over Eurasia. So we identified now the dog. There's a preprint as well published where they identified a sheep, which is carrier of these early strains. But we still lack the rodent population, because the rat, which is later on bringing Yersinia pestis to Central Europe, is still not present in the Mediterranean area, and not as well in Europe. They are coming more with the Roman Empire and spreading from there more or less to the rest of the world. But here we don't know the rodent population, and as well not an effective vector which can bring the Yersinia pestis from the rodent population towards the humans. Exactly, this is what I summarized here. So it's a bit older than expected. So up to now, there was always the hypothesis that we had just more with the first, second, and third pandemic, a thing which is playing in the last 2,000 years. The early strains are not flea transmittable, so we don't know the effective vector, more or less, or the vector host system of these early strains. It looks more that the early cases are zoonotic rather than epidemic outbreaks. So we think it's more a kind of single events which are happening over a long-lasting period in Eurasia. And later on, with the development most likely of bigger populated areas, maybe densely populated areas, so urbanization, then Yersinia pestis became more able to be spread by this rat flea vector system. If we have a look on other pathogens detectable in ancient DNA record, this is a short list, more or less, which kind of bacteria and viruses are well studied in ancient DNA context. We see that we have a big lack of these bacteria and viruses, especially in the time period where neolithization happened. And this is typical, the time period between 7,000 before present up to roughly 3,000 before present. There we see don't see any bacteria or virus which can potentially cause a bigger outbreak. So most likely, most of these infectious diseases which are circulating in this time are single events, zoonotic events for sure, which can happen and lead to a die of 1 or 2 persons. But we don't see this typical big outbreaks, which we know from later periods from medieval time. This is different, especially for the time frame where big urban structures are established, so around 3000 before present, when we see the shift towards Bronze Age and Iron Age, where we have denser populated areas, especially in the Mediterranean areas where we have huge cities established. There, for the first time, we can detect certain pathogens appearing or calculate back the most recent common ancestor, which are falling then in these time frames, like for leprosy or tuberculosis. We had as well then a look on the human immune system, if we see maybe adaptation ongoing of these of certain systems. And there it's important to know that we can more or less establish our modern day European gene pool by 3 big ancestral components. The so-called steppe population, which is incoming around 3000 before Christ, roughly. Then we have the early farming communities, which are coming slowly from the Near East and moving towards Central Europe and appearing in Central Europe around 5000 BP. And then there's the local hunter-gatherer population, which is incoming more or less after the last glaciation and building the more or less endogenous population of Central Europe. So between these 3 big components, we can more or less plot all the modern-day Europeans in their diversity, and we're carrying different proportions, more or less, of these 3 big components. Interestingly, in Europe, we can see a kind of gradient or first admixture happening between the incoming Neolithic population from the Near East with these local hunter-gatherer population forming a kind of client between these 2 big populations. And this was one of our biggest studies where we examined really early farming communities and later farming communities and compared them on the HLA level. Interestingly, we found that a lot of variants are declining drastically over time and some are rising in their frequency. And this was really astonishing to us because normally HLA variants are known to be balanced in the population, so they don't exceed too high frequency and they are quite variable in the population. So this is as well the HLA hypothesis, that you need a broad variability of HLA to be protected against most of the infectious disease. And this was really different in these early farming communities, especially where we see extremely high frequency of certain variants, which were unexpected. And we see as well frequency of alleles which are nowadays associated with certain autoimmune disease. For example, HLA-B2705, which is highly associated with psoriasis or ankylosing spondylitis. On the other hand, we see after this Neolithic period, when the steppe population is more or less admixing with these late Neolithic farmers, that we see a rise of certain alleles which are as well associated with autoimmune disorders. So, for example, DQB1, O201, which is one of the driving forces in celiac disease, or this DRB1 1501, which we heard already with leprosy, which is one of the major risk factors for MS. So here it's a bit tricky, and we didn't have the final conclusion why these autoimmune disease-related HLA alleles are on the rise. But what we can see for sure in the Neolithic population is that they had an atypical HLA dataset, which is not really well adapted to a lot of pathogens. So most likely they dealt only with a few and not with these major outbreaks or the diversity which we can detect nowadays in bacteria and viral world. We then had a look as well on some of the immune-related factors and genes, and one is from a particular importance because a lot of companies are producing biologics in the moment for this variant. It's the so-called protective variant in the IL-23R receptor, and this 23R receptor is modulating more or less our immune response, especially to the gut microbiome environment. So it's located in the IL-23 immune cascade, and in the end, there's the Th17 immune response, which are activated. And this protective gene variant, more or less, is depleting or down-regulating the immune response to this dysregulated microbiome. And therefore, it's as well a strong protective effect to certain autoimmune diseases, especially in the gut environment. And one of these diseases is ulcerative colitis or Crohn's disease, which is really a big upcoming modern disease. And therefore, it's a big target as well for development of biologics. We then had a look in the frequency distribution of these protective variant over time, and you can see it here in the upper plot, where we have the different populations. So it starts with the Western hunter-gatherers, so the local endogenous population in Europe, then the Anatolian farmer and early farmer, which are coming from the Near East, which showing here already a dramatic shift from more or less 0 towards 20% in the human populations. Then we see a constant slow decline of this variant, most likely due to admixture with the Western hunter-gatherer population. Then the steppe population brings in more or less a low frequency again. Most likely they had the same frequency like the hunter-gatherer population, and then we can see that it's still present in low abundance, more or less, in the European population. This indicates two things. So we see a dramatic increase, which we can only explain by most likely selection for this variant. So the people in the early farming community needed this variant more or less to be more protected against an inflammatory environment. And the constant behavior of this variant imply that we don't see big selection events later on working more or less on this variant. And this we tested as well with so-called EHH analysis, which gives you an idea more or less if a haplotype or a variant is selectively or selected in this time frame. And we can see here these extended haplotypes more or less appearing already in the Anatolian farmers, which are indicating that this was really the time point of selection. And afterwards, the selection pressure is not any longer existing that strong. This is as well reflected more or less, or these admixture events is reflected in the modern gradient of this variant over Europe. So you can see here in dark red colors the highest frequency more or less, and in lighter colors the lowest frequency of this variant in Europe. And you see more or less that the admixture events, so the out of Anatolia movement of the early farmers, represent one of the higher gradients. And the incoming steppe population more or less represents the lower frequency. And we see this climb more or less from northeast towards Spain, so to southwest, where we have higher frequency of this protective variant. And this is nicely correlating as well with the incidence rate of IBD, so the chronic inflammatory bowel disease. And we can assume more or less this protective variant has really an effect of modern day disease causes and distribution or epidemiological data. Why do they need it in pro-inflammatory lifestyle or an anti-inflammatory response of our own immune system? So this IL-23R variant, why it's needed more or less in these early times? And most likely, as I said, it has to do somehow with the dysregulation of the microbiome. And two big factors which directly came normally into mind is the first thing that they started to digest or to take in more milk products and dairy products, especially the problem is that we don't see a big adaptation to milk consumption in total. So the typical LCT loci, which we know, which is under high selection as well in European populations. So their ability to digest milk, sugar is coming much, much later as represented in this plot. So you can see here that in the last 2000 years, really this variant is positively selected. And the Europeans gain more or less this ability to digest lactose in the last 2000 years and not much earlier. So what worked really in early times is most likely more alcohol or fermented products. And this is one of the variants which is playing a major role in the alcohol digestion. And it stays already for 12% more or less of the ability of Europeans to digest alcohol. And you, unfortunately, you have to think now reverse more or less, because the variant which is plotted is more or less the variant which don't allow us to digest alcohol. And you see here that the hunter-gatherer population depicted in green was not really able, with at least with this variant, to digest alcohol. And it's a dramatic shift to 50% more or less in early farming communities. So it was a short time period where they gained the genetic ability to digest alcohol more or better in the total. And this is interesting because we see in the archaeological record as well a lot of traces for producing fermented products, especially bread, or maybe as well beer production. And one good example is here the Göbekli Tepe from Turkey. This is one of the earliest monuments or sedentary monuments we know from human history. It's dated roughly around 12,000 before present. And there are even experimental archaeological approaches ongoing where they started to brew a kind of rudimentary beer in the stone vessels. And you can see it here on the right side, and it should have even tasted quite well. So it's plausible that with the introduction of new foodstuff, maybe fermented products, we see as well this dysregulation of the gut microbiome, and we need a strong response more or less of our own immune system to downregulate the immune answer more or less to this new foodstuff. I didn't do this work all on my own, so I have to thank you a lot of colleagues, and I have to thank you for your attention.
Thank you very much. Well, that was a great presentation. And for the Q&A session in the room, I will ask Marin if he could. We cannot hear you. You have to unmute there, and I will look. If you want to ask a question, please raise your hand. Yes, Mihai, please. We'll start. You have to unmute. We cannot hear you.
Okay. It's all right now.
I wrote the question on the chat.
Yeah, I briefly saw it.
Can you read it? Yeah. How virulent were the earliest strains, Ellen, of pestis, C. pestis, comparing to the medieval ones? Does it support the theory that the start of the Jamnaya migration might be related to a plague epidemics?
Yeah, this is an ongoing debate, I have to say. So we are on the part that we claim that these early strains only cause single zoonotic outbreaks and were more like a chronic infection. And there's the Copenhagen group, which is claiming that we see this dramatic epidemic outbreak already maybe in the pre-Jamnaya integration phase. So they claim more or less that Yersinia pestis wiped out a lot of, or a big proportion of the European population, and that makes space for the Yamnaya integration later on. There are a couple of problems with these. So we don't know the actual rodent vector, to be honest. So we don't know how Yersinia pestis really were transported over Eurasia. Then the second is that we see these infections happening as well in the area of the Yamnaya population. So we see it overall. So Yersinia pestis didn't make any difference between the late Neolithic population in Central Europe or the Yamnaya people. So they should have died in the same manner, right? The third point is that we don't know the transmission route of Yersinia pestis between the humans. So they claim that it's the lung-based form of Yersinia pestis. If it's really this form, we know from medieval reports and early modern reports that if it's a lung-based form or transmission, then Yersinia pestis is killing quite quickly. So it kills in 2 days. And it's not a plausible factor that somebody from the steppe, for example, brought in 2 days Yersinia pestis to Central or Northern Europe. So this was quite too quick to really be transmitted effectively between these small villages. As I said, these early strains are all lacking the YMT gene. So as well, this flea transmission is not given. So there needs to be either a different insect vector, maybe, which is transporting Yersinia pestis, or there's maybe, as we identified the dog and another group, the sheep, there's maybe other animals which are bringing more or less the disease from the rodent population to the humans. But if this is the case, then it's more a single event and sporadic events than really a bigger outbreak. There's this paper as well from Gökhem side where they could show that different generations are infected with these early strains of Yersinia pestis, which is interesting. They identified it in a burial where the people were buried in the normal way, more or less. So they had really time to bury their people. They were not in a rush. So most likely they didn't die in one incident, but they died over longer time periods, which are indicating as well that the people most likely lived for a longer time with these infection. So it could be as well that Yersinia pestis maybe caused more chronic infections than really an acute outbreak, more or less. So this is what I think is the most plausible thing, that we had this zoonotic spillovers in this time, but that Yersinia pestis was able to really kill in a dramatic short way, more or less, the people, and that we have to deal here more with a chronic infection, maybe rather than an epidemic outbreak. And this is as well with the timing, as I said, a bit problematic. We don't have any indications of bigger mass burials, for example, from this time frame. We see overall a population decline, and then the Yamnaya population is incoming. That's true, but we cannot explain it with these patterns of Yersinia pestis. So from an epidemiological way, and from as well from the genetic way, it seems that Yersinia pestis was not that virulent like later on in the medieval time.
Yes, Consuel, thank you very much. Consuel, you had a question. Please read it.
Can you hear me?
Yeah.
Okay. Hello. Very, very interesting topic and extremely vast. My question, one of them is posted there regarding the 4 or 6 digits for HLA to detect traces of selection. Do you find it sufficient? I know there is a limit right now in terms of, you probably use the Illumina Dragon analysis or something similar to that to extract these, which is automatic and very nice. But 6 digits, I think it's a little better. But now I don't think we have the means to do it. So that's not more of a question. It's just something that I wish for. The second one, I have some data on contemporary genome and exome analysis. Do you recommend any pipeline to extract this, to get the hotspots for looking for this kind of data? We have some patients that are immune, there's some immune problems, so we suspect it's either contemporary things or something else more systematic in their genome. So we want to make this distinction between them. So is there any good, reliable pipeline that you could use right now to get these hotspots?
Oh, well, yeah. Maybe first commenting on the technical with HLA. So we are, I think, in the moment the only group who is doing really this in-depth HLA typing for ancient individuals, and this is especially the problem. So most of the modern day HLA typing is either done by PCR-based methods. So this is the classical way, or they are imputing the HLA variation based on these exome capture data or SNP chip data. And this is okay for modern day population, I have to say, but for ancient population, there are some limitations. So there are some TECSNPs which are working quite well, for example, for the DRB 1501. But for other variants, we are still lacking good TECSNPs. And that's why we developed a real capture for this HLA region to enrich more or less all the reads which are falling in this variability. And we have a more manual approach to call really the HLA variation. We developed an own pipeline which called Target, which is good for type 1 and type 2. There's only one technique in the moment out. It's called OptiType, which is still only available as a kind of preprint, which can be more or less call as well quite reliable HLA variation, but only for type HLA type 1. So there is really some limitation for low-covered shotgun data for HLA classification and calling. That's why we are limited as well for the 4 digits. The coverage in the ancient genomes are not that good that we can go further down or extend more or less to 6 digits, which I would love. I totally agree that as better and as more precise as more conclusions we can draw as well. But it was already fascinating that we can see so much variation and so much difference on this 4 digits level, which I found really surprising. The second question, good question. We see that more and more for sure from coming from the ancient DNA world, we're using the standard pipelines as used in modern genomic analysis as well. And we are working quite fine with them. To do a kind of selection, if I correctly understood, you wanted to do a kind of selection screen over the whole regions or over the whole exome. Yeah, there's not a perfect tool in the moment, I have to say. So we are trying as well with with low resolution, for example, with sliding window approaches of a couple of thousand base pairs, for example, or an FST analysis to just get some glimpses if there is some signature behind it. But there's not the perfect tool. And we struggled as well to find a good tool to indicate single selection events in the genome. So there we are, as I showed, we are ended up with EHH, which is not perfect for, especially for selection events which are not that strong in the genomes.
Okay, yeah,
But
it's okay. We cannot hear you, Consel. Yeah. Okay.
Thank you. Thank you.
Yeah, there is a follow-up question from Mihai. Mihai, can you hear me? You can put your question.
Yes, I can hear. I wrote it on the chat also. It's about the beer and the experiments with beer from Göbekli Tepe. So Laura Dietrich wrote in that article that the beer has no more than 2% alcohol in it.
Yeah.
So will it be enough to cause digestive problem for the ones who don't have the gene for digesting alcohol?
Oh, that's a good question. I actually don't know. I could imagine that as well more the dysregulation, so the availability of the starch-rich products more or less and sugar-rich products is causing more digestive problems than the alcohol level itself. It's interesting that these variants, which makes us able to digest alcohol, is as well on the rise at the same time. But for sure, this could be just an incidence somehow. So I don't think that the alcohol itself driven this. But we know that the subsistence strategy really changed from a more diverse nutritional basis. So you had a bit of cereals, you had a lot of other nutritional things, and a bit of meat as well. Changed to a more plant-based diet, and most likely as well a diet which is not that rich of fats any longer, which you need as well to take up some of the essential nutrients more or less in plant-based diet. So we see
probably the plants that they use to make that beer, the different plants used can cause different problems, different digestion problems. Because I know they use at least 2 types of plants for making the beer at Göbekli Tepe, at least from the experiments.
Yeah.
Okay.
Yeah, but it's hard to really pinpoint only the beer. But it's for sure intriguing, more or less, to take the beer and the alcohol, because it's as well a kind of socializing factor, and maybe a factor which played a role in general to get sedentary and to adapt this lifestyle in an environment which was really rich. So they lived more or less in a kind of paradise environment. So they had warmer climate time periods. They had a lot of games which they could hunt. So it's totally unclear why they started to construct these sites, right? Which for sure is then intriguing if you say, okay, they started to brew beer and to get a lot of tests, for example, and celebrations, and get there for sedentary. But it's yeah, it's for sure speculation.
Thank you very much.
Thank you. Now, if there are from the room, you can use the microphone, which is the omnidirectional, and only turn the laptop towards the speaker. If that,
There's no more questions from the room.
Okay.
Okay. So we're staying online then. Consuel, you or
Yeah, I have another question regarding the topic that Mihai has raised. So my question is, why limit ourselves to lactose, persistence in adults, which is the proper actual name of this thing, and why limit ourselves to alcohol, which has complex problems because it's linked to social aspects. So I think there is not enough coverage in the world for specific populations that somehow develop tolerance to different types of substances based on the local availability. There is a theory, I mean, starting with Darwin's finches, that if you live in a specific place, you develop adaptation to the local type of environment, and resources are very important. Now, speaking of lactose tolerance in adults, that's clear. You have young newborns that you have to supply, and they survive. And we're speaking about Neolithic domestication of animals and so on. But the same pressure can apply to regions. For example, I've lived for 2 years in China, and I've eaten rotten food. I mean, really something that, or toxic things that I really got really sick from them, because I probably don't have the means to deal with them. So there is very little study on these things, and also microbiome adjustment to this kind of topic. So this is a very unexplored area that could benefit and provide additional explanation, and especially an amazing a new perspective on how quickly can a population adapt to specific resources. I mean, rate of adaptation is something that's not very studied. It is not done only by selection. You die and survive the ones. It's also done by epigenetics. It's done by a lot of other things that have not been considered. It's probably not something that you would go in archaeology to get out, because it's complex. But it's something that we have to think about and then explore this data and then apply it, in a historical perspective. That's just more of a comment on the whole thing.
Thank you for the comment. It's exactly in the line what I would say. For sure, especially in this basic research from the ancient DNA, we cannot do this, right? So we even have more sparse data than in modern day research. And as you said, there are a lot of things understudied, which are highly important, more or less, to understand the selection which is ongoing in these ancient genomes, or even which is predisposing us, for example, in modern day disease. I totally agree. And for sure, LCT and the alcohol digestion, these are simplistic things which are commonly available, and where at least a couple of studies are published. And even there, I have to say, the studies are really limited to certain fractions and also to certain populations, which is highly problematic. And what I can show as well with this ancient data is that we be really cautious with the interpretation or the transfer of studies which are done, for example, in Europe to Asia, for example, or to Africa. So because they had a totally different population genetic history, So they had different influences which shaped their modern day genomes. That's why it's super important to do these ancient DNA studies more or less as well in Asia and in Africa. And I would really love to see more data upcoming, to see maybe events which are similar to Europe, or which we can parallelize, for example, in Africa, where the milk production and digestion as well come much later with the Bantu expansion, for example, in the last 2,000 years. It would be interesting to see if similar genes or regions are under selection since this time, for example, or where we can see as well the introduction of domestic rice in Asia, if this has the similar effect like the introduction here of the domestic plants in Central Europe. And this would be highly important to understand how selection and evolution is ongoing in our human, or in humans in total. We see as well that most of this selection is not working really on single variants and single genes, I have to say. It's more a polygenic adaptation really to certain environments. So we see more small effects which are summing up in total. This is as well the case for IBD. So we see not only this IL-23R variant under selection, but almost 100 variants which are more or less acting in a concert, which are dysregulated or regulated in the beginning of this Neolithic period. So there is something which looks like a selection coming from this phenotype, but for sure this phenotype was not present in the ancient population. So this is a modern disease phenotype, which we cannot one by one transfer more or less to Neolithic population. So there is a lot of things ongoing which we actually don't understand, and where we have to go more into a broader sense and get as well more information from other sources. What could hint to this nutritional basis is as well modern studies on T cell variability, especially in this disease phenotypes, for example, in IBD, which could show that T cells in IBD patients is as well recognizing different yeast types. So it points as well into this direction of fermented food products or alcohol production, that there is as well an overreaction towards this, and maybe as well a connection between intake of yeast and the development of IBD later on. So it's much more complex than we thought in the beginning. It's for sure not only based on this variance and mutation, but as well on hundreds and thousands of different factors, most likely. But thanks for the comment.
Thank you. Other questions online or from the room? I could put only two small questions detail that I had during your. One was already answered, as Mihai already pointed to. I think the one because you said that there was not the FLIA who played a role in transmission, right? And I was curious of what other hypotheses are. For this, you mentioned it, and the second was to clarify more. I didn't understand. It was a bit too quick. The link between leprosy and the MS today, right? You mentioned it a bit quick, and I would like to know more detail.
So this was this slide. So we did this first case control study, and as you see from the publication date, it was 2018. So it was the time where not this big genome-wide data was available. And from modern day genome-wide association studies, there are different susceptibility variants known. And the strongest one, which is modern day associated as well with leprosy, is really this HLA-DRB1 1501 allele. And this allele is as well quite well known in MS studies, where it's the strongest susceptibility factor. And there was last year a publication in Nature as well, where they could show that this allele was most likely really brought with the steppe population into Central Europe, and causing more or less as well the modern day gradients of MS over Europe. So it's a bit similar to our IL-23R story, but there they showed it for MS variants in total. As well, for MS, it's for sure not only DRB 1501, but it's one of the major factors which is leading more or less the high score. And it's interesting that it plays as well in leprosy a big role in the susceptibility. But as I said, as you can see here in the last bar of the chart, that this variant is not vanished completely, more or less, from Europe. So it's still present up to 15%, especially in Northern Europe, where we see as well a high ancestry of the steppe population. So it's still there, and therefore most likely there's a pleiotropic effect. So it doesn't have only negative effects, more or less. So in this case, it would be the problem that you are more affected by leprosy and MS, but it should have as well some kind of protective effect.
So in Northern Europe, you mean that would be, you claim that after the 16th century that it was that decline, right? But not in Northern Europe, or I mean, I was just visiting last year the leprosy museum in Bergen, and that was, let's say later, right, over the last three centuries, right?
Yeah, that's true. In certain areas, there are still minor infection rates for leprosy detected. It's as well present still in Greece. There's as well the spina longa, for example, which is, I think they run it until the 70s. So there are leprosy infection, but not in this broad epidemic outbreak that we know from medieval time, where a lot of people were really affected as well. So as I showed with the anthropological record, that we see 5% of people really have signs and severe signs of leprosy. Yeah, it's true.
And 100% infected. That was really.
Yeah, exactly. So everybody was in contact, more or less, and this is the open question still with leprosy. So we don't see any big changes in the genome itself. So there's no loss of virulence, for example, in the 16th century. We don't see a big adaptation of the human immune host system, most likely indicated by this HLA variant, which is still there. So there's still the susceptibility of modern day population, more or less, to leprosy. But what we know from modern studies is more or less that Europeans, even if they are coming into contact with leprosy and leprosy patients, they never get the disease. So it seems that our immune system is so potent, more or less, that we can outcompete this bacterium nowadays. And this is most likely just to the living conditions of the Europeans, or due to the living conditions. So this is as well a factor which we cannot measure by genetics, more or less, which is playing a more, or having a higher impact, more or less, than the genetic basis.
So it's no comparison with other continents or other.
Yeah, and this is why most likely in Brazil and India we have these higher incidences, because the living conditions are just more problematic. So the immune system of the people is more compromised, more downregulated, most likely, and more susceptible, therefore, for the disease. And this is as well another study which I didn't present here, was about an early medieval cemetery where we see a high infection rate of different diseases, and that was really astonishing that we see multiple infection and a lot of diseases which are just present in a small community. And interestingly, this community was buried shortly after the small ice age, which appeared in the end of the Roman Empire, and where we know that most likely the people were male or had malnutrition or problems with the nutritional basis, and were therefore more susceptible, most likely, as well, for different infectious disease. So most likely this factor, like misharvests, problematic nutritional basis, plays a big role in the appearance of these epidemics.
Thank you. Yes, if there are other questions online or from the room, right? Who's in the room? If not, then we thank you so much for a very interesting and nice and consistent talk, and wish you good luck, and we'll follow your work and keep in touch. Right?
Yeah, exactly. Keep in touch, and if you have a follow-up question which appears later, drop me an email, contact me.
Yes, wonderful. Thank you. Thank you very much.
Thank you. Bye.
Bye.